Thank you, Christoph. Hola, buenos dias. That is about the limit of my Spanish, if that's any good. So I will save your ears now and apologise in advance for sticking to English. But yeah, thank you very much. Yeah, I'm going to give you really a bit of a stroll through Seychelles amphibians and some of the other taxa that are there already. So nothing too strenuous for a Thursday lunchtime. So, introduction to Seychelles amphibians, 63 million years in the making. So, I always start off with my acknowledgements at the beginning of a talk. Lots of people leave it to the end, but I like to put the organisations and the people that have been key to all of the work that I've been able to do at the beginning of a talk. So I'm not going to stand here and list out all of their names, so don't worry. But just to highlight, yeah, there's been lots of organisations and individuals in organisations that have been very helpful in the work that I've been doing. So yeah, many thanks to them. And thank you also for asking me to come and deliver some, hopefully some new information for you on Seychelles amphibians. So lots of people seem to think that when typhoons and hurricanes occur, when I tell people that I'm in the Seychelles, they seem to think that it's in the Caribbean. It's not in the Caribbean, as you can plainly see, just in case. In the Western Indian Ocean. And the main islands upon which I've been working are the inner granitic islands of Mahe, Silhouette and Pralin. But there's something pretty special, really, about Seychelles. So this is a representation of the planet around about 200 million years ago. The area that we're interested in is here. You might sort of recognise that shape, and it will all become clearer very shortly. So what we can see here now, the direction of the arrows are representing the shifts of tectonic plates, and the colour of the arrows represents each individual plate. And we can see former supercontinent of Gondwanan now starting to break up. What we see here is an India-shaped block of land which is slowly moving northwards. Seychelles is round about here. So give or take a few million years or so, roughly 66 million years, or roughly 63 million years ago I should say, the Seychelles broke off from what became continental India and were then isolated out in the Western Indian Ocean. And the inner islands, the granitics, have never actually been fully submerged. So there are outer islands, coralline atolls and such like, but the inner islands have always remained. So the islands that we currently see, the inner islands in Seychelles that we currently see, are basically the mountain tops of what was previously a microcontinent. So this block here was a microcontinent of about 129,500 square kilometres. So quite a considerable landmass. Obviously being isolated and never having been fully submerged, there are endemic fauna and flora that have persisted there, or their ancestors have persisted there since that breakup took place in the isolation in the Western Indian Ocean. It's only recently that we've really started to look at the biogeographic relationships of taxa on Seychelles. So, as you might expect from oceanic islands, there's quite a few reptiles. There's a couple of species of snake. This is Seychelles house snake, very closely related to the house snake that you'll find on mainland Africa. There's quite a variety of species of geckos as well, actually. This is a Seychelles sucker-tailed gecko. As you can probably imagine, it's called a sucker-tailed gecko because there's a sucker on its tail. It's kind of a little bit prehensile, and it uses it to support it when it's clambering around in caves and trees and among branch roots and its habitat in Seychelles. There's two different species of Phelsuma, so some of you are probably familiar with Phelsuma from Madagascar. Seychelles has got their own two species of Phelsuma, day gecko. There's also chameleons. Initially there were two species described. One was kind of forgotten about, And it now looks like that they are, at the very least, two very distinct morphotypes that occur on Seychelles. And there's some work ongoing looking at the morphological and genetic differentiation in between these two chameleon populations. There's also a variety of skinks, so Mabuya, or rather previously Mabuya, now Trachylepis. These guys are all over the place. If you're out doing field work, they will happily watch you while you're turning over soil in the hope that you're going to be turning over nice little grubs and insects for them to dive in and nick as well. So I've been out going through leaf litter looking at frogs, and there's been 2 or 3 skinks sitting there waiting, looking at all the leaf litter that I'm turning over, waiting to dive in. And they're quite bolshy little characters. They don't really care. They're quite tough. There were previously three species of tortoise as well. Only one now remains, the Aldabran tortoise. So I mentioned about the biogeographic relationships that haven't really been properly explored. And this is a good example of some recent work looking at some of the gecko species, the Aeolianic species. Essentially what we can see here is that there does seem to be quite clear groupings from these two species here, Aeolianic seychellensis and Aeolianic tachyscopius, and they seem to be clustering in groups and also specific to islands. So there seems to be some differentiation happening here. And it's also worth bearing in mind that we don't really fully know the cycles of emergence, connection, reconnection between the intergranitic islands. There's been lots of volcanic activity in Western Indian Ocean, glacial periods. It's very difficult to actually nail down particular times. So we don't really know these cycles of connection and reconnection, how and when that they've happened, but they've undoubtedly had an effect on a lot of the species there. Anyone know what this is? Apart from Christoph and apart from Ivan.
Cecilia.
Yes! Well done, that man. Good. Did anyone not...
About this way, I can call you.
No, yeah, I mean, could be, yeah, close, but no cigar. Yeah, has anybody not heard of caecilians before? So 3 orders of amphibians, anurans, frogs and toads, chordates, chordata, newts and salamanders, and then the gymnophyomans, the caecilians. And Seychelles is quite a special spot for caecilians. Up until 2017, there were 6 nominal species that derived from an endemic radiation on the inner granitic, or that remain on the inner granitic islands of Seychelles. Superficially, as you can probably tell, they're very, very similar. They're pretty difficult to tell apart. The sorts of things you have to look at are the annuli, the rings around the body, whether they're subdivided or not. You can also look at the distance between the eye and the tentacle and the nose. But I mean, you think snakes are tough and muscly. You pick one of these guys up and they're slimy as well, because obviously they prefer the moist habitat. They're very, very difficult to wrangle. So although you can say, oh yeah, I can count the rings, and oh yeah, I'll just look at the distance between the eye and the nostril, when you actually try to do it, it's pretty tough. It takes a little bit of practice. So yeah, superficially very similar, and they only really occur on the larger granitic islands. 2 recently described species, increasing the number of Seychelles chameleons. So Hypogeophis petit, probably the smallest caecilian in the world, around about 10 centimetres. And it's known from just a very few localities, just on one single island. Second, Hypogeophis montanus, again, just known from a single island, and again, just from very few localities. There seems to be a sweet spot on and around Mont Seychellois, which is the tallest mountain on Seychelles. And at the moment, they just seem to be found in just a particular altitudinal band, and nowhere else on the islands. There's also a tree frog. One thing that's pretty remarkable about these, so this is Tachynemys seychellensis. Its closest relatives are the hyperoleid tree frogs that you'll find on Madagascar. The interesting thing about these guys, you might think that transoceanic dispersal in amphibians doesn't really happen very often, being that amphibians don't really go for salt water very much. But these guys probably rafted over from Madagascar about 11 or so million years ago. A lot easier if you're a tree frog to actually raft over, rather than if you're a terrestrial frog or a frog that lives sort of closer to the ground. All you need is a massive mudslide or a big area to fall into the sea, and as long as you don't get in the water, fine, you can stay on that for ages. And as you're all probably aware, amphibians and reptiles can persist for quite a long time without feeding. Although probably within the little tree islands that were possibly rafting across at certain points, there were a variety of other organisms in there. So the journey could have been quite easily sustainable by other invertebrate prey that were transported at the same time as these frogs. Has anyone seen these before? Good, I'm glad you have, Krista. The Nazikibatrachids, so quite a special group. In 2003, they were discovered, or one species was discovered on the western slopes of the Western Ghats, also known as the purple burrowing frog or balloon frog. In 2017, another species was discovered on the opposite side of the Western Ghats, on the eastern slopes of the Western Ghats. And we think that their origin was probably around about 66 million years ago, so just about the time that Seychelles had actually separated from that Indian subcontinent landmass as it was moving northwards. The reason why these guys are important, and I'm talking about them, is that they're the closest living relative to the Sooglossidae, which I'm now going to introduce you to. So the whole family is endemic to Seychelles. And they're fully terrestrial, so no waterborne tadpole stage. Their whole life cycle takes place terrestrially, and they don't enter water at any point in their life cycle. And it's the family is comprised of 2 genera, and there's 2 species in each genus. I'll just give you a brief introduction to them now. So first up, Zooglossus seychellensis. Anyone make out what's going on in this picture? Yeah. So this is a female, and she's carrying 8 little metamorph froglets on her back. So there's the head of one there, head of another one there, head of another one there, another one there, another one there, another one there, another one there. I think there's 7 in total. So what they do, the male crawls from a nice little damp patch, hoping in vain that a female frog will find his damp patch enticing. And they get on and do their froggy stuff, deposit the eggs. The eggs are guarded, so one or possibly both of the parents actually guard the eggs while they're sitting there. The tadpoles develop inside the egg, but then the tadpoles hatch out of the egg and climb onto the back of the female. And then she hops off to carry on doing her normal froggy day-to-day stuff. So parental care is evident right from the offset. So from egg deposition right all the way through to metamorphosis of the young. And through that whole period, the tadpoles don't feed. So they're called what's known as endotrophic. So the eggs are really quite large proportionally and very, very yolky. And that sustains the offspring all the way through their development until they hop off the back of the female way after the tail has been fully resorbed. That's a little metamorph sitting on the edge of a 10-pence piece, which is probably ever so slightly larger than €1. How I managed to find this individual, I don't know. I think it was more luck than judgment. But I got to take this really nice photo, so I always stick this in because I'm very proud of it. But you can see that they're really quite small individuals. and the adults are only, on average, about 1 centimetre snout vent length. They can get up to about 1.5 centimetres, but usually around about 1 centimetre. Sister species, Zooglossus thomaseti, as you can see, this is much, much larger. Snout vent length about 6 times the size of Zooglossus seychellensis. Interestingly, and some of you may be aware of this already in any case, quite often even the larger Anuran species still have very small offspring, and these are directly comparable to the size of Zooglossus seychellensis offspring as well. Oh, and I should have, yeah, in the bottom corner I've sort of highlighted that all the zooglosses are edge amphibians, so evolutionarily distinct and globally endangered. I put their edge ranking there, and also their IUCN, their assessed IUCN red list status. So Zooglossus seychellensis on the previous slide is currently currently endangered, and Zooglossus thomaseti is currently critically endangered. So onto the second genus, Seychellophrine. These were previously thought to be the smallest terrestrial vertebrate. Average snout vent of round about 1 centimetre, around about 10 millimetres. That title has now been taken by the pedophrine frogs of Papua New Guinea. Average male snout vent length of about 7 millimetres. So even smaller than these guys, but that's an adult sitting on my finger there. They still carry out parental care, but it's a little bit of a different process. So this is a female here that's just deposited some eggs. You can see that they are pretty large eggs and very, very filled with yolk. But the whole developmental stage takes place within the egg. So you can see on here there's arms, legs, heads. It's quite nice. We've got a comparison here of two different clutches. But they go through the whole tadpole stage, the whole developmental stage into a tiny metamorph froglet, and then they hatch out of the egg. But the female stays with the clutch all during that time. And they are also on the edge list and currently recognised as endangered on the IUCN red list. Sister species, Seychellophrine pipilodryas. Superficially very similar to Seychellophrine gardneri, around about the same size, although on average a little bit larger, so about 1.2 millimetres snout vent length. They tend to have, or some of them tend to have this really sort of dark reddish brick coloration, and they have a hastate mark on the back, which is kind of like an arrowhead. Although interestingly, I found gardneri on 2 islands where this frog doesn't occur that also have this hastate mark. But I haven't found gardneri on the same island where this frog occurs that have the hastate mark. So it may be a tentative characteristic you can use to differentiate, but it might only be valid on when you're differentiating between gardneri and pipilodryas on the one island. So zooglossidés, 4 species. Zooglossa seychellensis, find this one at Mahe silhouette, thomaseti, Mahe silhouette, gardneri, Mahe silhouette, pipilodryas, only found on silhouette. However, in 2009, anyone heard of Valley de Mai? Apart from Ivan, possibly. So the Valley de Mai is a World Heritage Site on the island of Praslin. It's basically a palm forest. Predominated with coco de mer, the largest seed-bearing or the largest seed that you'll find, kind of a double nut. Anyone come across those? Okay, have a look on the internet. Good. So there's a World Heritage Site on Praslin, and one of the organisations that I've been working quite closely with, Seychelles Islands Foundation, they are responsible for this World Heritage Site. And they were actually in the process of building a visitor centre. In the construction of that visitor centre, somebody spotted a frog jumping around in some leaf litter and material at the back of where this building was being constructed. And then it was subsequently identified as being a zooglossid. But no one had ever reported zooglossidés from Praslin before. The only frogs you'll find on Praslin, or up to that point, the only frogs that were thought to be on Praslin were tacininus, with a big tree frog. So it raised quite a few questions. How did they get there? How long have they been there? Why have they been missed before? What's the story if they have been there for a considerable amount of time? How are they related to different populations on different islands? So Sooglossidae seychellensis now appears to be on 3 islands. And that's kind of where I come in. So field work, as you can see, was pretty tough. It's very, very hard work. There's never any alcohol. And it was, I can't lie. Seychelles is a great place to do field work. However, you do have to climb that. So it's nice when you get back down the bottom, when you can have a beer and catch up with people, walk into the sea and have a pina colada and all that. But you do have to climb up there, and it takes quite a long time. This is the sort of habitat that you come across. So as I've said previously, the inner islands are granitic. So you get these big granite boulder fields, and the frogs, they love it in there. So you're walking along all this stuff and then diving in and trying to catch them and hoping that rocks don't crush you while you're doing so. So occupational hazards. Fortunately, obviously, I haven't been crushed yet. Good thing. When you get up to the top on the 2 main islands, Mahe and Silhouette, this is the sort of scene that you're greeted with, even when it's 33 degrees down on the coast and all the tourists are having a nice time. This is what it's like when you get up to the top, and it's quite stunning. Even on a hot day, you get up there and it's cold, or cooler anyway, and it's a pretty special place. Virtually every surface is covered with moss and lichen. And there's frogs and caecilians knocking around all over the place. So it's pretty special. That's the view from that previous picture when I'm looking up the mountain, taken from down there. This is a bit like a holiday snap, so sorry about that, but here's a nice view. Up the top there, it's a little bit sparse and open, but things like pitcher plants, which are always cool to go and see. Lots of diving in rock crevices, trying to catch frogs. Zooglossid field work, you get very acquainted with everyone's arse, because that is essentially what you get to see most of when you're trying to find frogs, and also caecilians. And hopefully what happens when you come out of underneath the rock, you've got a frog in your hand, which I have there. Everyone knows what this is like. Something really important to point out here, local rum, very nice, called Takamaka, gets you through the night, excellent. On an important note though, the other arrow here is pointing to some slides, which are really the reason why I'm here today working with Christoph, because we've got some blood slides in there that we're looking at. So these are the actual slides. It's not just random slides, they are actual slides that I've got here today. Exciting, yeah? Okay, good. So some of the work, some of the output that we've had just recently, so I've mentioned about the biogeographic relationships, and thinking back to the previous slide where we had the geckos. The frogs also seem to be pointing us in a certain direction, in that the different populations on different islands appear to be evolutionarily distinct lineages. So the silhouette frogs all clustered together and appear to be a sister taxa with the praline frogs. The Mahe frogs are a sister taxa to a clade comprised of the other two. So each island population of Seychellensis has got its own evolutionarily distinct lineage. Then the same again when we look at Tomasetti. Silhouette population, Mahe population, very clearly defined and well supported. Same again with Gondwanan, silhouette and Mahe, and then obviously we have Pipilodryas at the top, which is only found on one island. So this has obviously got implications for conservation. So previously thinking about conservation for 4 species, now we need to think about evolutionarily significant units and protecting lineages and making sure that the evolutionary trajectory is, at the very least, supported when we're thinking about conservation action and intervention. So one of the other things which I'm currently working on are the vocalizations. So any of you that do work with anurans are probably aware that anuran vocalizations, advertisement calls, are species specific. So when we have a look at the vocalization of frogs from Mahe, it's a complex call. So a lot of frog calls aren't complex, they're just a brr, brr, brr. Good. Glad I got at least a little laugh then. I would have been crushed inside otherwise. Zooglossidés, or two of the zooglossidés, make complex calls. So this is one note and then a series of secondary notes. So this is a primary note and these are secondary notes. We can see here there's a build-up of energy. It's kind of a bit like winding up an elastic band and then letting it go. And then that peters out and then that's followed by 4 primary notes. Hopefully I've got my laptop turned up loud enough so you're going to be able to hear this. No. Okay, we'll skip that bit. But you can clearly see the structure of this call. Yeah?
Do an impression.
I got to now because I've already done a non-complex frog call. So it's kind of like a... If you want to send me a text message when we've finished, it's my ringtone for text messages on my phone. So we can revisit this. Having a look at praline frogs. So first thing you notice about this is that it's decidedly longer. Many, many more secondary notes. I've heard up to about 21 secondary notes. So just to remind you again, these are the secondary notes. But then when we look at the construct of the primary note, it kind of fades in. It's more like a... I should have brought some of that rum with me. It would be much better. But you can see that the energy in this call is quite evenly distributed all the way through that primary note. So it's very, very differently constructed. So we're looking at, we're looking at candidate species, basically, pending further work, at the very least for Zooglossus seychellensis. Anyway, so I mentioned conservation. Fortunate enough in 2017 to get an MBZ, or to get MBZ funding to establish some long-term monitoring. No or very little conservation work has taken place on Zooglossids and indeed on caecilians and all Seychelles amphibians historically. So we wanted to try and establish some long-term monitoring protocols on the 3 main islands where we find Zooglossids, but also extending that to some of the other islands where you also find, or where you find caecilians where there are no Zooglossids, but you do also find tree frogs as well. So one of the things that we've done is purchased SN4s. Has anyone used song meters before? Yeah, excellent. I share your pain. Yeah, pain, yes. Sometimes programming can be a bit of a nightmare, as you can see from Francois here, very sadly holding his head in his hands. But we've got a series of these units, and they've now been established on praline, on silhouette along an altitudinal gradient. So the idea is that we'll be able to monitor, remotely monitor zooglossid activity, and also use that as a bit of a proxy for caecilians as well, because they do tend to be sympatric. Zooglossids themselves are really difficult to study. You have to search for them. You can't just walk around and see them hopping around, or not generally anyway. So the only real way you can find out if they're in an area is to listen out for their vocalizations. So we've got a number of these units that have been set up to record 5 minutes every hour, 24 hours a day. Once a month, Francois and his team go and change the batteries, swap the SD cards, get back down to the office, load it onto a flash drive and stick it in the post. Because internet in Seychelles is prohibitively expensive, or still is at the moment. So the easiest way to actually send data from one place to another is just stick it in a flash drive and bung it in the post. I receive it, say I've got it, download the data, send it back. So I always like to point out Saeed, because that was after we climbed up the mountain. You can see how happy he is about having to climb in sort of pouring rain for 4.5 hours so we can put up these SM4 units. Francois and Tisha are happy, and that some of you might recognise her. That's Karen Lips. So she's we're very fortunate to have Karen involved in the project, giving us some advice on policy matters and also helping us with global knowledge of amphibian disease and disease ecology. So yeah, very grateful to have Karen on board. So all of us are happy, apart from Saeed, but he's generally all right. He's, you know, don't be put off. He's a nice guy. The other thing that we've been doing in tandem with setting up the SM4s or the song meters is deploying data loggers. So these things here, tiny tags, they're recording temperature and relative humidity, and they're going to be able to allow us to look at our behavioural data, i.e. the vocalisation data, and see how that relates to temperature, relative humidity, and changes in temperature and relative humidity, and also in relation to where the units have actually been put along an altitudinal gradient. So this is Simon Maddock. He's doing lots of Caecilian work. We work quite extensively together in Seychelles. Again, as I've said, a lot of the data that we're going to be collecting and are collecting will also serve as a proxy for Caecilian work as well, because they do overlap in habitat. Because Caecilians basically you have to dig for them. Although on a couple of places, or a few places actually in Seychelles, they're so abundant that they're probably the most vertebrate biomass that you will find on an island, or even in a particular given area. And you can sometimes see them swimming around in streams at night. When I was there recently, we were, again, it was chucking it down with rain. It was almost like a torrent coming down the trail, but Caecilians were out on the trail. And as you're walking up, you're treading on snails and all sorts of stuff. You come back down and you find the Caecilians eating the snails. So yeah, quite exciting. Not exciting for the snails, obviously. So we've been working with a lot of local partners. This chap, Lindsay Chong-Seng, you probably recognise from a few slides ago, he's been instrumental in all the work that I've been able to do in Seychelles. And this is Jess. She's just finished her undergraduate degree, or last year in fact, at the University of Seychelles. And what we've been doing here is taking disease, or doing disease screening for Seychelles amphibians. So taking swabs for BD and B-cell, the two potentially lethal chytrid fungi. Right, can anyone hazard a guess at what NGO, or what sort of NGO, these people are from? There's loads of clues in the picture. Anyone? To give you a clue, what's he holding in his hand?
Beach volleyball or something.
Flip-flops.
Oh.
Yeah. So this is the Marine Conservation Society. They wanted to join us on some of our, so they could learn about disease screening amphibians, which you might think is a bit weird. However, MCSS have been strategic in some conservation work that they're actually doing at a lowland freshwater wetland site adjacent to a beach. So it kind of overlaps with their remit. And there are Caecilians at this site. So they wanted to know from us, so they came out with us to do some disease screening of amphibians. I don't know what would possess you to think that going into the forest halfway up a mountain wearing flip-flops is necessarily appropriate. Bearing in mind there are lots of palms around and they've got big spikes coming out of the bottom. Anyway, they were good, and yeah, we found quite a few amphibians. So we've got a Caecilian, just there. So yeah, we took MCSS out and did lots of amphibian disease screening with those guys as well. So just recently, we've established the same protocol on Praslin Island. So establishing song meters and deploying our data loggers. And the hope is in the future, when we get some more cash, we'll be able to buy more units, but the capacity would have been built already by the NGO and the people that we're working with on Silhouette, and the NGO and the people that we're working with on Praslin, so ICS and SIF, and also Seychelles National Parks Authority and University of Seychelles. All of these will be able to work together to deploy the same approach on the main island, Mahe, hopefully at some point in the not too distant future. So we're already starting to see some interesting patterns. So you can see here there's a very clear and different relationship between temperature and humidity just at one particular site on two islands, and that obviously has a bearing on amphibian ecology on those islands. Other things we've been doing, conservation needs assessments. Has anyone come across conservation needs assessments? So if you're working with amphibians, this is a really good method by which you can basically feed in situ and ex situ data or lack of data on the species on which you're working, and that will basically produce a set of conservation recommendations for you. So we've done it with the 4 currently recognised species of Sooglossidae and also all of the caecilians. And it tells you things like in situ research would be good, ex situ research would be good, conservation education would be good. I've put these 2 frogs here as well. So these are the least concern, and that's an introduced frog. But these are found quite commonly, very low down. These are around people's houses all the time. So they're very good proxies for raising awareness of the risks to amphibians. Also good proxies for possibly developing captive husbandry skills in country. And also raising awareness of conservation issues. Just also because these are found a lot lower down, people are aware of what's happening with these frogs that might actually signal a change if there's been an impact of climate change, temperatures increasing, or possibly if disease has arrived. We would potentially see it in these guys before we saw it in the Sooglossidae or in the caecilians. So there's still quite a lot that we don't know. Taxonomy is still very much being worked out for all of the species. We don't really know distribution limits. We don't know if there have been previously local extinctions. We still only have limited information on reproduction and life history, and conservation threats to disease susceptibility is a big question mark as well. But hopefully the monitoring that we're going to be doing is going to be acting as an early warning system for the risks posed by climate change, and also regular disease monitoring is also going to act as a bit of an early warning system as well. So if we stop hearing frogs in a particular area where we're doing some recording, that might mean something's happened. So we need to make sure we get out and do some swabbing basically to see if there is something going on in terms of disease. So yeah, thanks to Christoph and Ivan. That's the reason why I'm here today. Thank you all very much for coming to listen to me waffle about Sooglossidae. Thank you very much for listening. And because there's lots of dead stuff usually knocking around, I thought it'd be nice to see something that's alive, doing what frogs do. Cool. Cheers. Thank you very much.